Okay. Yo, I'm here to talk about the work that we've been recently doing on digital staining. And I'm going to just start a little bit about the motivation for our work. And then I'm going to talk a little bit about the kind of how it applies to the kind of 3D cellular systems and organoids. So as you folks know, we build these wacky microscopes that are label free. So this is to address some of the problems with fluorescence imaging. Some problems include, toxicity, fixation, preparation time. You need to develop the tags. If you want to screen a lot of cells, fluorescence is typically slower. So one of the questions that I want us to think about as we're going through this is what has more information? Does the label-free data have more information, or does the fluorescent image have more information? Okay. So I'm kind of going to begin in the middle of things and talk about this computational specificity approach we've been developing. So I was interested in this procedure called EBICS, where you inject a sperm into an egg. One of the questions that we had was, how do you tell which sperm to inject into the egg? So we ended up scanning these microscopy slides that we had the known outcomes for. And we tried to figure out which sperm were good and bad. The problem was that when you scan in so much data, you have to somehow analyze it. So I think this is a problem that a lot of us, face. So we tried to go with the neural network approach for image segmentation, which was based on this unit architecture. So the input for this the input for this architecture is, one of our label-free phase images, and the output is a label head, neck, and tail. Okay. So this was the training for this was done. We basically annotated a lot of images, expanded our annotation set. Then we did some fine correction, and then we had a classifier that was ready to analyze our box of slides. This is kind of fun. What we found here was that there are certain parameters that are correlated, but in absolute terms, these parameters are small. In biological terms, they're mostly to do with hydrodynamics or the preservation of certain internal cellular components. So all this work was fun, but the problem that we really had here was how do you avoid annotating all the data? So we were thinking about where we can get specificity from. And it ends up that a very good place to get specificity is the chemical specificity afforded by fluorescent labels. So this is this kind of digital staining work that we're doing that we're now calling PICS. So the idea here is that you will run your time-lapse microscopy experiment using our label-free quantitative phase imaging. At the end, you will fix your cells, and then a neural network will train how to reproduce the fluorescent stain, and then you can digitally play back that stain. You can digitally fix your time course experiment. So this approach works very well with some of our optical instruments, where we essentially have co-localized fluorescence on the next channel. So to do this work, we use these deep convolutional neural networks to perform this back propagation. And I really like this image here because it hints, it kind of illustrates the new features and new, basically the difference between neural networks and some of the more classical techniques, which I mentioned here. But one of the important things is that the neural networks, like the one we're using, UNet, they will essentially learn both the texture information, how bumpy the cell is, but also contextual information, which you can see when it's highlighting the edges and such of these cells. So this picture here on the left is our phase image input, and on the right is the operations that transform it into the synthetically stained GAPI output on the far right. Okay, so this technique that we developed is basically works for everything pretty well, or it's pretty widely applicable. So we can do different stains, we can do different cell lines and different modalities. So, of course, as we did this work, we learned some stuff. One of the things that we learned was that there is a relationship, especially between the fluorescent image quality and the resulting synthetic stain that we get. One way to address this is to get really nice fluorescence, which we now should be able to do as we have acquired some confocal equipment. An interesting thing, if you compare the synthetic stain versus the actual stain, so a lot of stains work really well, but some stains aren't that uniform. You can see here that some area, you can see where my cursor here is, there are some areas, okay, yeah, one sec. Yeah, so there were some areas that the images were not stained compared to the physical stain, although this picture is gone. But one of the important things here is that by using our quantitative phase imaging microscopes, because they decouple the phase from amplitude information, we essentially are able to get a much better synthetic stain compared to like a regular DIC or phase image or bright field image. So in this technique, what we essentially do is we acquire co-localized fluorescence and label-free images and use that to train. One question might be, how many co-localized label-free and fluorescent images do you need? The interesting thing is that we found out you don't need very many of them to get pretty good results. So we did some computational experiments, and essentially, after only maybe about 5 minutes or so of training on a really nice GPU cluster, maybe like 20 training pairs, you can get pretty good synthetic stains. So of course, this stuff is real time. Here's the DIC. You switch to our glimpse system. Perform computational integration, which is part of our now reconstruction process. Here's the hardware physical DAPI stains. And then if we go here, we can see the synthetic DAPI stains that we did. So this enables time-lapse playback of otherwise toxic stains. So this whole, hello? So with this experiment here, the cells were never stained, and we just apply our neural network and see what kind of results we get. It looked pretty reasonable. You can basically do a lot of single cell analysis this way. The DAPI and DIL essentially, or sorry, DAPI and DII essentially enable you to divide the nucleus from the cell body. Oops. Okay, so we're kind of going a little bit fast, but one of the questions that I wanted us to think about early on was what is more information? Does the fluorescent tag have more information, or does the scattered light label-free image have more information? Well, it ends up that the fluorescence obviously tags for a very specific thing, but in that way you do lose some information. So we tried running this digital staining stuff, just for fun backwards. You can see that obviously you can't go from DAPI back to the label-free image. So there is some loss. Okay, so let's talk about 3D cellular systems.
Sorry, really quickly, we have just a couple more minutes. Maybe I'm going to say like 3 at most. We start opening it up for questions.
Yeah.
Just FYI.
Yeah. So I have 2 more slides. Okay. So this is basically some of our preliminary, although we submitted these to a journal, results on working on 3D cellular systems. So you can see the rightmost column is the physical DAPI stain. The middle column is our attempt to make a synthetic DAPI stain of these spheroids we got from Dr. Jun Kong's lab. One of the things that we did here is we compared the essentially the nuclear fraction, how much nucleus versus cytoplasm there are in the cells. We compared the hardware DAPI versus the synthetic one, and we found that they were only really about 3% off. So I really do think that many of these questions that we have about about characterizing cells can be answered with these kinds of artificial and synthetic stains. Okay, so to have a discussion, a little bit of a discussion. So this is kind of what we're thinking for our future work, or at least I am. In this work, in the work that I've shown so far, we've mostly focused on using two-dimensional neural networks. But in principle, the neural network, in the same way, can eat an image and spit out another image. It can eat a cube and spit out another cube of data. So I think that especially for the spheroids that we're working on, we're getting a lot of stuff being messed up due to focusing effects. So I think that probably in the immediate, one of the things that we're going to do is transition some of our neural networks to three-dimensional neural networks. It isn't that bad, but it will consume a lot of computing resources. So is labeling the cells based on the fluorescent image. So once you produce the synthetic fluorescent cells, that's kind of good, but you need to ultimately binarize the individual cell mask to, for example, determine the image I'm showing on the bottom is like a cell cycle state, a very common one. It's this Fusing reporter. And one of the big problems here is that even if you have the fluorescence, you somehow need to positively ID the cells as being in any of the parts of the cell cycle. It's a little bit harder than it looks because if it's ratiometric, it's based on how much the cell's expressing, not the total. So digitally establishing thresholds is a problem. So finally, one of the grander challenges is to perform what's called instant segmentation, where you throw in the label free image and you will output every single cell, annotated individually. So there are some classical methods to do that based on watershed, but now more end-to-end machine learning approaches such as MaskRCNN are coming up. One of the issues is that you really need to have very high resolution images to individually segment cells. Okay, so that was a fast talk, went over a lot. I think I'm ready to take any questions or discuss some of the material. Thank you.
We have time for just to give enough time for the final presentation.
So that was great. Let me ask you, so here it seems like you're mostly dealing with one kind of cell type and, it may have different features.
No.
Okay, so you haven't mentioned that. So can you clarify that?
Yeah, so first off, the neural network is trained at the end. So it basically works for any type after it's trained. Some of the images I've been showing were, so we've had CHO cells as W cells. We were trying, sorry?
Within a single image or single, so I mean, what I was alluding to is that you can train it for the different cell types. My question is more with respect to as you get to organoids, for example, and as these systems evolve with many different cell types in kind of a three-dimensional space, where these cell types kind of interact, evolve over time.
Okay.
So can you elaborate on that a little bit? And for example, imagine that the organoid is developing over the course of a few weeks where you want to monitor what's going on, then I imagine you can't just do like, a single endpoint staining, that you'd have to really understand, the development along the way too, and make sure you capture the intermediate cell types as well.
Yeah, I think that's, I think there's some truth in that. But one of the interesting things that we've been finding, especially with machine learning, is that some of the features that the neural network is able to pick up on are not immediately obvious to people. So I think a good example here is this slide here. So if you take a look at the top panel, that's at time 1, or at the beginning of the experiment, and the bottom panel is at the end of the experiment. So at the end of the experiment, training, and then after the experiment's over, we do this, stain and training procedure, right? So you can see that actually there are not many fibroblast-like cells at the end of this experiment because we didn't passage the cells and they got angry at us. But the neural network was still able to stain fairly well this change in morphology. So I think that, or this different fibroblast-like morphology. Also, this particular experiment involved a mixture of SW cells. You have these big SW480 and 620 cells. So I think at the end of the day, this approach is fairly robust.
Ron, I don't think it was clear. This is a mixture of two cells here. And
Can you repeat that, Gabby?
before, and actually, if you run the movie, the network is picking up these two particular cells, no problem.
Mm-hmm.
During the differentiation, of course, that's a much harder problem. It would help if we could train a priori on individual differentiated cells, maybe. Do you think that's doable? Ron?
Are you asking me, Gabby, or I mean, yes, I mean, it'd be interesting to see whether you can, for example, let's say, take an organoid and actually dissociate it.
Yeah.
Right, and then that dissociation somehow, I don't know, maybe separate the cells at that point, or you know, they lose, you know, I don't know how, you know, their form changes obviously once you dissociate them. So I'm not sure, you know, what the correlation would be with the dissociated terminal cell types and the ones that are adherent to one another in like a, you know, intact organoid. I mean, it's a good question.
Or the same terminal ones can be used
Yeah.
data for machine learning.
The stained terminal within the organoid.
Within the organ.
Yeah, and then the question would be whether you know, one of my questions was, you know, given the high degree of transformation in you know, morphologies and things like that, that one terminal stain would be sufficient, and you really have to capture kind of the intermediate states as well as the cells are changing lineages and so on. And, you know, like I'm actually relating that a little bit to Jose's presentation, you know, where we, you know, there's that notion of like the pseudo time, right? So that pseudo time is dealing with internal markers of gene expression, but you can imagine that those also correlate to, you know, cell morphology. Right? So the question, to what extent can we correlate these two talks, right? Where you can also establish something about, you know, pseudo time and pseudo cell types along the way.
Is it possible, in a kind of brutal way, to fix organoids, stagger them in time, and then slice them, fix and slice, and maybe that would be a good representation for training?
Maybe, yeah. We actually did. We have a 3D slice.
Parachain embedding.
Yeah, yeah. We actually have data we can even maybe share with you.
So,
Yeah. Oh, we should probably move to the next.
yeah, we need to